Hello there, everybody. Today we're going to be looking at another positive piece of evidence that supports the truthfulness of the idea that the Earth and universe are roughly 6,000 years old. And this piece of evidence we'll be looking at today is the existence of soft tissue inside of dinosaur fossils, and in particular inside of a Triceratops fossil that Mark Armitage found back in 2012. But before we get into what Mark Armitage discovered, we need to give you the necessary background information leading up to that. So, I will bring to your attention this article put out by NC State University News on November 26, 2013, titled, "Iron Preserves Hides Ancient Tissues in Fossilized Remains." And the article says the following. New research from North Carolina State University shows that iron may play a role in preserving ancient tissues within dinosaur fossils, but also may hide them from detection. The finding could open the door to the recovery of more ancient tissues from within fossils. Mary Schweitzer, an NC State paleontologist with a joint appointment at the NCU Museum of Natural Sciences, first announced the surprising preservation of soft tissues inside a T-Rex fossil in 2005. Her subsequent work identified proteins in the soft tissue that seemed to confirm that the tissue was indeed T-Rex tissue that had been preserved for millions of years. But the findings remain controversial in part because no one understood the chemical processes behind such preservation. And I'm going to tell you why they don't understand the chemical processes behind such preservation. Because you see, according to the evolution atheistic paradigm of history, dinosaurs died out millions of years ago, long before human beings came into the picture. And so there shouldn't be any soft tissues whatsoever inside any dinosaur fossils or bones. And yet, in 2005, Mary Schweitzer found soft tissues inside a T-Rex fossil. This was so controversial, her discovery, that it almost destroyed her career. But her discovery could not be refuted, and so they accepted that she had indeed found soft tissue inside a T-Rex fossil, which should be impossible according to the evolution paradigm of history. And so they had to create an alternative explanation for how these soft tissues could survive for millions of years inside dinosaur fossils or bones. And the reason why they had to find this alternative explanation is because if you don't, what you end up dealing with is the fact that the existence of soft tissues inside dinosaur fossils and bones opens up the door to the idea that human beings and dinosaurs lived together at the same time at some point in the past, which, by the way, is taught by the Bible. And because the academic and scientific communities are controlled by God-hating losers who don't want anything to do with God, they're going to reject the idea of dinosaurs and human beings living together at some point in the past as hard as they can. And so they're going to try to create an alternate explanation for how these soft tissues can be preserved within fossils and bones for millions of years. So what was their alternative explanation? Let's keep reading. Schweitzer's latest research shows that the presence of hemoglobin, the iron-containing molecule that transports oxygen in red blood cells, may be the key to both preserving and concealing original ancient proteins within fossils. Her results appear in Proceedings of the Royal Society B. Iron is necessary for survival, but it's also highly reactive and destructive in living tissues, which is why our bodies have proteins that transport iron molecules to where they are needed but protect us from unwanted reactions at the same time, Schweitzer says. When we die, that protective mechanism breaks down and the iron is turned loose on our tissues, and that destructive process can act in much the same way formaldehyde does to preserve the tissues and proteins. Hemoglobin seems to be the key. Both birds and crocodiles, the dinosaurs' closest living relatives, have large nucleated red blood cells. Therefore, they also have more hemoglobin per cell than mammals. If dinosaur blood cells were similar to either one of these species, which seems likely, then their blood cells would also contain much more hemoglobin than human cells, amplifying iron's preservative effect on the tissues. If the hemoglobin were contained in a bone in a sandstone environment, keeping it dry and insulated from microbes, preservation becomes more likely. Schweitzer and her team noticed that iron particles are intimately associated with the soft tissues preserved in dinosaurs, but when they chelated or removed the iron from soft tissues taken from a T-Rex and a Brachiosaurus, can't pronounce that, the chelated tissues reacted much more strongly to antibodies that detect the presence of proteins, suggesting that the iron may be masking their presence in these preserved tissues. They then tested the preservation hypothesis by using blood cells and cells taken from modern ostrich bone. They soaked some of these vessels in hemoglobin taken from red blood cells while placing other vessels in water. 2 years later, the hemoglobin-treated soft vessels remained intact, while those soaked in water degraded in less than a week. So what Schweitzer and her team did was they took cells from ostrich bones, they put some of those cells in water, they soaked the other cells in hemoglobin, and after 2 years the cells that were soaked in hemoglobin were preserved, and the ones that were in water, they degraded in less than a week. So how did the legacy media react to this news? They ran with it, baby. CBS News said, soft tissue found on T-Rex explained. The Huffington Post said, controversial T-Rex soft tissue discovery finally has an explanation. And NBC News says, what preserved T-Rex tissue? Mystery explained at last. Mary Schweitzer has finally provided us an explanation for how soft tissue can survive in dinosaur bones and fossils over millions of years. But that's where Mark Armitage came in and rained on their little parade. First, I'm going to let Mark Armitage explain some of the problems with Mary Schweitzer's experiment regarding the hemoglobin-soaked ostrich bone cells. In the video titled Iron 2A, Critics Who Defend Iron Preservation of Dinosaur Tissues, we see Mark Armitage say the following at the 15-minute and 53-second mark of the video.
When you look at Schweitzer's explanation, her theory that iron preserved the tissues, and you study the materials and methods section, which is a little difficult to find, but if you go and find it and you study it, you realize that Dr. Schweitzer had to first use an anticoagulant to prevent the blood from clotting. Why did she do that? She put an anticoagulant in the blood that was used, the chicken blood and the ostrich blood that was used to soak the tissues in, because if that blood coagulated, it would have bound up the iron and made it unavailable for her experiment. She needs the iron. She has to have the iron free and available for the experiment. And so she first has to put an anticoagulant in there to prevent it from clotting, number one. Number two, when you read the materials and methods, you find that she used ultracentrifuges multiple times during the experiment. Why? Well, she had to filter out all the serum that was full of clotting factors and other factors that bind up these proteins tightly in the clotting reaction. She had to remove the serum with all those clotting factors. She had to remove all the cells, all the platelets and subplatelet cells and other cells, white cells related to clotting of blood and of tissues to form scabs. All of that had to be removed through multiple series of ultracentrifuge use. And finally she had to use molecular filters to filter out these things. And so she spun everything down multiple times, and finally she was left with the red blood cells, which she broke open. It's called lysed. She lysed them open, and that, of course, freed the hemoglobin, which has the iron in it. Now, none of those conditions that can be said are indicative or related to what happened at the Hell Creek. Okay? When those things were buried at Hell Creek, there were no ultracentrifuges. There was no anticoagulant available. There were no molecular filters. And so just to get to the point where she could begin the experiment, she had to employ all that laboratory technology. And then, of course, she left it on a laboratory bench at room temperature for 2 years in the absence of water, which is abundant at Hell Creek, the freeze-thaw cycle, the heating of the Montana summer, and all of the microbes and scavengers and insects and everything that was fully involved in those specimens, and particularly my specimen, the Triceratops horn, fully open to the environment, highly vascular, and so she had to remove all that from her experiment in order for her experiment to work.
That's right, guys. You heard that correctly. In order to get her experiment to even work, Mary Schweitzer basically had to employ a bunch of controls and filters that would not have existed out in nature. That should tell you that her theory is probably wrong. Of course, Mark Armitage didn't just simply come along later on and debunk her study. No. He actually found something in 2012 that completely destroys the entire explanation that Mary Schweitzer and her team created and that the legacy media ran with. On January 3rd, 2013, Actica Histochemica put out a study by Mark Armitage that was peer-reviewed, made it through the peer-review process, got published in a secular academic journal related to this field of science. And the study by Mark Armitage is called Soft Sheets of Fibrillar Bone from a Fossil of the Supraorbital Horn of the Dinosaur Triceratops horridus. Now, if my pronunciation of scientific names is bad, please forgive me. So that's what you see here as the title of the article. And when you skip to the materials and methods section, you read the following. An intact Triceratops horn, HCTH00, was recovered on May 12, 2012 from a well-sorted fluvial sandstone within the Hell Creek Formation at a previously unexcavated site on a private ranch within the Hell Creek Formation. And then he gives coordinates for where he found it. The recovered horn was jacketed and removed. The length, girth, and external morphology of the fossil was consistent with other Triceratops horns recovered from the Hell Creek Formation. Disarticulated Triceratops ribs, HCTR11, and vertebra, HCTV22, found within a mile of the horn were also recovered for analysis. Hand-sized pieces of HCTH00 were fixed in 2.5% glutaraldehyde solution, buffered with 0.1 milligram, sodium cacodylate, buffer at 4 degrees Celsius for 5 days, rinsed in distilled water, and buffer and stored in phosphate buffered saline, PBS. Individual pieces of roughly 20 centimeters squared were removed by pressure fracture, so with his hands. HCTH010203 examined under a dissecting microscope and probed with sterile forceps to identify and collect soft material. Soft materials recovered were washed in double distilled water and stored in PBS awaiting further analysis. Other horn specimens, HCTH0405, were processed through a decalcification protocol. Several pieces, about 20 to 50 centimeters in size, were rinsed in double distilled water after fixation and were incubated in a solution of 14% sodium EDTA at room temperature. EDTA was exchanged every 2 to 4 days for a period of 4 weeks. Significant bone mineral remained after 4 weeks, therefore it was unknown whether complete decalcification would yield soft and transparent vessel-like tissues, such as previously reported. And then he references Mary Schweitzer's work. So when Mark Armitage found this Triceratops bone in the Hell Creek Formation in 2012, part of the bone was fossilized. But the non-fossilized part of the bone had soft tissues in it. So this whole iron acting as a preservative method, or theory I should say, that Mary Schweitzer came up with was predicated on the bones being fossilized. Yet here Mark Armitage found non-fossilized Triceratops bone that had those exact same soft tissues in it that the T-Rex fossils of 2005 had. So Mark Armitage's discovery completely destroys the sales out of the whole iron acting as a preservative theory that Mary Schweitzer came up with the next year. And so as you can tell from the date of this article, Mark Armitage put out this study like a full 10, 11 months before Mary Schweitzer ever came out with that press release outlining her theory. So the issue of soft tissues inside dinosaur fossils and bones is still a fatal flaw in the evolutionary atheistic worldview, because the evolution paradigm of history tells us that soft tissues inside dinosaur fossils and bones should not exist, and the one theory they came up with to try to explain why they do just doesn't work, because we have found fossilized and non-fossilized dinosaur bones that contain these soft tissues in them. And the iron as a preservative theory doesn't work with non-fossilized dinosaur bone. And in case you think I'm misrepresenting what Mark Armitage said about the non-fossilized specimens he was working with, I'll let him explain it himself in his YouTube video, No EDTA Hornbone, where we see the following.
Hi folks, Mark Armitage here with a brand new video that I've not yet put up on YouTube because I didn't think it necessary. What you are watching is me peeling soft fibrillar bone from the deep inside core center portion of the Triceratops horn. And the reason I have to put this video up online is because I have all of these, well, what should I call them? Ignoramuses, ignorant people who refuse to be taught about how demineralization of bone takes place using EDTA, a weak acid like vinegar. It's like soaking a bone in vinegar. And they are convinced that it is the EDTA that is soaking into the bone and actually softening the tissue. Well, here it is, folks. This is bone that was not soaked in EDTA. It's from the dead center of the Triceratops horn, and you see me peeling off all this soft fibrillar bone. So the process that scientists use to demineralize bone does not soften the bone. It's already soft. Bone, by definition, is a soft tissue. It is mineralized, and we use EDTA to remove the minerals. And so for all these folks who are, unbelieving Thomases, here is your evidence that this is already soft before we processed it.
Well, there you go, folks. You got Mark Armitage on video himself showing you the soft tissues on the Triceratops bones that did not have EDTA applied to them. It's right there for you, in your face. The very evidence that evolutionists don't want you to know exists, the one thing that completely destroys their entire worldview, right there for you on video. It's not just some study in an academic journal that Mark Armitage did. You got it on video here. And, of course, the academic and scientific communities embraced Mark Armitage's discovery with open arms, attached to middle fingers. As we can see from this CBS Los Angeles article from 2014, lawsuit, CSUN scientist fired after soft tissue found on dinosaur fossil. You see, at the time that Mark Armitage made this discovery and was working on his study and the paper he submitted, he worked as a scientist at Cal State University, Northridge. And 2 weeks, roughly, after he published his findings in that paper in that academic journal, he was fired from his university. He was accused of bringing his religion into their school, onto their campus, even though, if you read his study for yourself, he makes no mention whatsoever of young earth creationism. And of course, this turned into a lawsuit that drug on for years until eventually CSUN decided to settle out of the lawsuit with Mark Armitage. Basically, according to Mark Armitage's attorney, if CSUN had gone to court and actually done trials and stuff with Mark Armitage, they would have been found guilty of wrongful termination. So rather than go through that embarrassment, they paid a very large settlement to Mark Armitage. As you guys can see from this video, the folks who control the scientific and academic communities are not interested in the truth about reality. The majority, and I would say the vast majority of the people in these communities are God-hating atheists and evolutionists who do not want people to discover anything that upsets their worldview. And if people do discover something that upsets their worldview, like Mark Armitage's 2012 discovery of soft tissue inside non-fossilized Triceratops bone, well, they're just going to make the evidence disappear and get rid of the person who found it. That's exactly how they roll. They were employing censorship against creationists before social media platforms like YouTube, Facebook, and Twitter decided to make censorship commonplace. And that, ladies and gentlemen, is positive evidence for young Earth creationism. Non-fossilized dinosaur bones containing soft tissue completely disproves evolution, billions of years, and the iron preservation theory that people use to explain soft tissue lasting for millions of years. But in the young Earth creationism worldview, soft tissue existing inside non-fossilized Triceratops bones makes total sense because dinosaurs and humans lived together at one point in the past.